So welcome to the part 2 session of your muscle physiology. To begin with, we have already spoken about the basic structures of your actin, myosin, troponin, tropomyosin. Then we spoke about what is the actin head, myosin head, how does the interaction occurs, what is the sarcoplasmic, endoplasmic reticulum, calcium, ATPase, how is your contraction occurs, how is your relaxation occurs, everything we spoke. To summarize, step 1, your calcium enters into the cytoplasm, binds with your troponin C, activates it at a plus 40 millivolt. Step 2, activated troponin C activates your troponin T. Step 3, troponin T activates tropomyosin. Step 4, tropomyosin moves away, exposing your myosin binding sites of actin. And step 5 is your myosin, it combines with actin and that causes a contraction. How is your relaxation occurs? There is a circa, then there is a ATP combining with myosin head, and this is how your entire process occurs. Now, during contraction, what are the things you need to remember? A band is going to remain same, whereas your H zone is going to be disappearing, whereas your I band is going to get shortened, and your sarcomere length is also going to get shortened. So, what is important here is your force of contraction is always depends on the length of scar. Okay, your sarcomere. So your force of contraction is going to be maximum only when the sarcomere is going to, I can't call it as a directly proportional. Your force of contraction depends on the length of sarcomere. When the sarcomere length is going to be at an appropriate size, only then the force of contraction will be maximum. That means even if the sarcomere is increased length, or there is a decreased length, your tension, or I can call it as the force of contraction, will be changing. That is what your length tension relationship tells. This relationship, this length tension relationship can be studied on a whole in a skeletal muscle preparation. What I want to tell you exactly here is the length of the muscle can be varied by changing the distance between the two ends of the attachment. At each length, the passive tension is measured, the muscle is then stimulated electrically, and the total tension is going to be measured. So the relationship of tension to muscle strength is going to be shown by this diagram. Okay, you can see here there is a percentage of your resting length, and here you can see the tension. Okay, so when this is going to be 100%, okay, that means that when the length is going to be increased between 100 to 120, there is going to be, at this length, there is a maximum overlap of myofilaments producing maximum number of cross bridge and maximum amount of tension only between this 100 to 120. When it is going to be decreased length, even then there is not going to be any formation of your contraction, or even when it is going to be exceeding 120%, even there is going to be exceeding 120% of normal sarcomere length, even then your force of contraction decreases. So the entire muscle as well as the individual sarcomeres, optimum length is required for the maximum tension. More or less than the optimum length is not going to yield you. So this is what Starling's law tells. The force of contraction is a function of initial length of muscle fibers only up to physiological limits. When it is going to be exceeding the physiological limit, again the force of contraction reduces, and when there is going to be a decreased than the relationship, that is also going to be relaxed. That is what, okay, that is what you're going to see here. There is going to be a sense organs within your muscle. Now tell me, now answer along with me. The length of muscle at which the force of contraction is going to maximum is called as optimum length. Or for a skeletal muscle, optimal length is resting length. The tension generation is maximum at optimal length because of all available myosin head will be presenting to the sarcomere only at the optimum length. The length of the sarcomere at which the tension generation is maximum is 2 to 2.2 micrometer. Now, this is the place where you need to understand the sense organs within your muscles that is going to be involved in the muscle binding. Okay, so you have a myocyte, isn't it? A muscle fiber. Okay, this is going to be your muscle fiber. So your muscle fibers are going to be covered by endomysium. Your muscle fiber is going to be covered by endomysium. Now you first again remember I told you the first thing. So first you're going to have a muscle spindle. So from the spindle you're going to be covered by the epimysium. So this epimysium are going to have numerous fascicles. So fascicles are going to be there. Multiple fascicles are going to be there. The muscle cells are going to be arranged as a cluster. I'll show you once again the first thing which we discussed. So this picture what we are going to talk about now. So you're going to have a epimysium that is going to be covering your muscle. Inside your epimysium, going to have numerous fascicles. These fascicles are covered by perimysium. Okay, these cells are created for contraction and generation of muscles. Okay, force. Okay, these going to be generating the force of muscle. Now, in order for a muscle to contract, you need to have a nerve fiber. You need to have a several nerve fibers that are going to be innervating. These nerve fibers, there are two types. So these nerve fibers, there are two types. One is 1B and second is 2. What is this 1B and what is this 2? Remember, inside a muscle fiber, I'll tell you, inside a muscle fiber, what happens is, okay, just consider this as your muscle fiber. Inside your muscle fiber, you're going to have two types of muscle fibers. One is called as nuclear bag fiber and second thing is called as nuclear chain fiber. Okay, so this is going to be one thing. Okay, so it is going to be like this. Okay, inside this, you're going to have a numerous like this, a chain of thing. Okay, from here, it's going to be again, there is going to be a sensation that is going to be released and this is going to be carried by. So whenever there is going to be a muscle relax, okay, whenever there is going to be any sensation, okay, there is a reflex one which is going to travel, take a sensation that is called as 1A. So this 1A is a sensory fiber. This sensory fibers takes the signal to the spinal cord. It's going to be taking a signal towards your spinal cord. So this is going to be a spinal cord. It takes a signal to your spinal cord. Okay. So actually your signaling will be going through your down. Okay. From there, what happens? Okay. From there, what happens? It has to take the sensation to the up towards your motor area. So towards your motor area. So remember always the upper part is going to be your sensory, lower part is going to be your motor. So what happens now? It's going to take inside the gray matter. This is going to be again the gray matter. This is going to be your center part. So to the gray matter, there is going to be taking 1A is going to take your sensation. From 1A, it has to travel back. It has to go down. From there, what happens is there is going to be a traveling back. So this sensation travels back through 1A, okay, A delta fibers, so A gamma fibers. So your nuclear back fibers are going to receive the sensation, to receive the signaling through A delta. Similarly, you're going to have, this is called as nuclear bag. Similarly, you're going to have other thing that is again going to be like this. So this is called as nuclear chain fibers. So this is going to be again nuclear chain fibers. In the nuclear chain fibers, what happens is the signal is going to be traveled by your signals are going to be taken by the other one. What is that called as? That is called as two fibers. We will be discussing again this about the muscle fibers also. So the information is going to be taken by two muscle, okay, fiber 2. Okay, this is going to be 2. And from here again, it is going to receive back. The receiving back is going to be again done by 8 gamma fibers. So the neuron start from the spinal cord, comes to your muscle spindle that innervates and activates your muscle spindle. They sense a change in the length of the muscle and forms a sensation. What is this called as? Proprioception. So this proprioception is the one that is going to carry through your 1A and 2 fibers into your spinal cord. From the spinal cord, after the spinal cord comes, what happens? The spinal cord are going to carry the signal through A gamma, and this A gamma is going to go into your muscle spindle and causes contraction. Muscle spindle and causes contraction. In the muscle spindle again, what happens? The sense organs that are going to be present in the muscle spindle, you call it as a Golgi tendon. So what happens? Your muscle spindle is going to be starting like this, and you're going to have a muscle like this. Okay, so this region is called as your spindle. This spindle region has receptors, and it is going to send the sensation through sensory fibers. What is that sensory fibers called as? Sensory fiber is going to be 1B. You saw 1A and 2 in the muscles. In the spindle, you're going to have 1B. This is nothing but your Golgi tendon organ. Golgi tendon organ. So the sensors are going to be developing. It's going to sense the tension that is developed in the muscle, inside the muscle. So muscle உள்ளதா வந்து உங்களுக்கு one A gamma is going to supply. This A gamma is also supplied to the Golgi tendon. So the Golgi tendon organ is a sensory receptor 1B that is also going to transmit the information of the sensation. Why all these are required? They wanted to interpret the and also create a balance between the contraction relaxation as well as the sensation. So entire mechanism is going to be controlled. Entire mechanism is going to be controlled. Okay, this is what I need to understand. And one of the important thing is, now you understood what is 1 fiber, 2 fiber, 2 is again divided into 2A, 2B, everything now we are mentioning, right? What are these things? We will be discussing this after completing the neuromuscular junction. Now what we are going to talk about is the neuromuscular junction. Why this neuromuscular junction is important, or why you need to talk about this neuromuscular junction? Because this is the place where there is going to be transmission of your impulses for a muscle to contract. That is why we are going to next talk about the neuromuscular contraction and the energy source. So this is what we are going to talk about. Okay, now before moving to that, you need to understand what are the different types of contraction, what are the energy sources, isn't it? So that is what we are going to discuss here. So different types of contraction you are going to see is, there are different types of contraction. Okay, either they can be isotonic or they can be isometric. Okay, either it can be isotonic or there can be isometric contraction, or there can be a different 3 types of contraction: concentric, eccentric, isometric. All these things are there. What are these types of contractions? I'll discuss now. So what happens is isotonic. What is the meaning of isotonic? So tension is produced in the muscle while there is a change in the muscle length. Okay, it is a dynamic contraction because the movement occurs. Okay, there is a movement occurs. That is why I called as isotonic. So what is the meaning of isotonic? There is a tone remains the same, whereas your length is going to change. In the length changes இருக்குல்ல, அதுல either your muscle length can shorten or your muscle length can lengthen. Muscle length shorten ஆகும்போது that is called as concentric contraction, whereas muscle length increase ஆகுறப்ப that is called as eccentric contraction. Clear? Then what is isometric? Here your tone is going to be changing. Okay, that is your muscle contraction is going to occur, but there is no change in the length. So the tension develops in the muscle, but there is no change in the length of the muscle. It is a static contraction as no movement occurs in the joint. How to remember the example for this? You're going to take a dumbbell. Okay, you're going to take a dumbbell. Okay, this is your elbow. Okay, this is going to be your arm. This is going to be your forearm, your elbow. You're going to hold a dumbbell like this. Okay, you're going to hold a dumbbell in your hand. But remember, you're going to hold the dumbbell, but there is no increase or decrease in the muscle length. It's going to remain constant. But what happens? There is a contraction that is called as isometric contraction. Clear? Okay. So the isometric contraction, the muscle of the upper limb while bringing the foot to the mouth by lifting weight, etc., the muscle length is going to be decreasing. For example, I'll tell you where the muscle length decreases. Where the muscle length decreases. So you're going to hold a dumbbell, but now what happens? You're going to flex your arm. You're going to flex your elbow. While flexing, what happens here? You're going to have your biceps. Okay, you're going to have your biceps. So this biceps, what happens is going to be normally what happens. This is your normal biceps. Here you're going to have the connection. Okay, so you're going to have a tibia, you're going to have a humerus, you're going to have a radius ulna. So it's going to be connected to your radius ulna, and you're going to have a biceps here, which is connected to the humerus, head of humerus. Okay, so here this is the normal length. But what happens whenever there is going to be a flexion, the muscle length decreases, but the tension in the muscle is going to remain the same. Okay, so you're going to lift a weight. So this is going to be here extra weight is produced, greater energy is used, but then your length is decreasing. So now coming to the isotonic muscle contraction. So isotonic muscle contraction example you're going to give, etc. Now coming towards the isotonic contraction. Isotonic contraction and isotonic contraction. Okay, again can be concentric, there can be eccentric. Eccentric na downward phase. So you're going to increase the length. Biceps brachii produces tension and it's going to be length and relaxing. No movement, na isometric contraction. I think I have clearly explained you what is isometric, what is isotonic. All these things can be drafted in a graph by the mechanism called as kymograph. This kymograph is going to produce the, it's going to give you the graphical representation. Now, in the exam, they can ask you a 2 mark or they can ask you a 5 mark on what is the difference between isotonic and isometric contraction. Isotonic tension remains the same. Muscle length can be either increased or decreased. Work is done. Load is going to be on CVS is going to be less, so BP will be reduced. Whereas isometric, increased tension, muscle length remains the same. No work is done. Load on the CVS will be more. Therefore, there is going to be rise in the BP. Okay, now coming to the energy source. You know that where is the energy source for the muscle contraction? Muscle converts chemical energy into mechanical work. Immediate source is ATP. Ultimate source is metabolism of your carbohydrate and your lipid. ATP hydrolysis occurs. I already told you in the last part during the muscle contraction and relaxation. Your ATP is stored in the muscle, initiates contractile activity and depletes in 3 seconds. How do you resynthesis your ATP? Because ATP is required. How are you going to resynthesis your ATP? So phosphorylation of ADP by creatine phosphate. Creatine phosphate plus ADP gives you creatine plus ATP. This is called as Lohmann's reaction. Again, I repeat. So creatine phosphate plus ADP gives you creatinine plus ATP. This is called as Lohmann's reaction. That is the reason why when the athletes when you're going to do a heavy exercises, immediately when you're going to take a creatine level in your body, there will be increase in creatine. Who is responsible? Who has given this? Lohmann's reaction. Now, glycolysis can also occur by and give you ATP. 38 ATP molecules can be given from one glycogen. So, glycogen converts into 2 pyruvic acid and 2 ATP. 2 ATP is going to be again occurring from glycolysis. Then what happens? This 2 pyruvic acid again goes to the Krebs cycle. Krebs cycle 34, 16 into 2, 34 you are going to get. So 34 plus 2 ATP நடக்கும் ஒரு glycolysisல. So இந்த pyruvic acid 2 pyruvic acid இருக்கு. 2 pyruvic acid 2 ATP produce பண்ணும் in glycolysis, and in Krebs cycle again 2 during Krebs cycle, 34 utilizing your plus H plus ions. Therefore there is going to be overall 34 plus 2, 36 plus 2, 38 will be there. Krebsல 34 were the 16 into 2 plus you get 2 ATP from glycolysis, then you get 2 ATP. So 34 plus 2, 36 plus 2, 38 ATPs you obtain from the glycogen alone. Then oxidative metabolism, 95% of energy used for total இவ்ளோ ATPs உங்களுக்கு வருது. This is what you need to understand. And ATPs எந்த அளவுக்கு required இருக்கோ, so oxygen demand is also very much necessary for a muscle. So oxygen demand is going to be directly proportional to the intensity of the exercise. It's going to be directly proportional to the intensity of the exercise. Okay, that you need to remember. So the volume of oxygen consumed during exercise, the maximum amount of oxygen that can be consumed while performing severe exercise. That is VO2 maximum. So oxygen depth maximum oxygen on sometime every time you won't be able to get. That time what happens during the oxygen depth, you're going to go into anaerobic pathway. So anaerobic pathway what it is going to be doing is lactic acid metabolism மூலமா oxygen is going to be produced. So at rest, so when there is a maximum work, there's oxygen deficit. So oxygen deficit time, what happens is there is going to be lactate anaerobic metabolism. After anaerobic metabolism, once again, when the oxygen is obtained, that is going to go to the recovery phase. Okay, pH, resting condition 7.3, onset of muscle contraction goes to acidic due to dephosphorylation of ATP to ADP. Later part, what happens? Once there is going to be resynthesis of ATP from creatine phosphate, alkaline medium pogo, but at the end of muscle contraction, you will be producing pyruvic acid, lactic acid, all these things moves towards acidic environment. So now coming to the thermal changes. When ATP is hydrolyzed, only a fraction of it is used for energy. Remaining all is going to be liberated as heat. This ATP hydrolyzes in muscle during contraction liberation is going to cause a heat production. So even at rest, what happens? It is the heat given off when the muscle is at rest, primarily due to the basal metabolic process that is going to be happening in your muscle. For a basic metabolism that is occurring in your muscle, that is going to releasing energy, heat, that is going to be producing that this minimal heat at the resting state, that is called as resting heat. Then you have an initial heat during muscle contraction, where all heat will be produced. Number one, initial heat is produced by activation of your muscle. Okay, initial rapid liberation of energy before action muscle to contract. So calcium ion is going to be releasing. There is going to be myosin, ATPase activation. All these things are going to cause initial heat. Then what happens? Your muscle is going to get contracted. Because of it, there will be a heat releasing. And there can be isometric contraction. That time maintenance heat will be there. Okay, then what is a recovery heat? Recovery heat abdina. So the excess heat following your muscle contraction continue for 30 minutes following the cessation of your muscle contraction. That is recovery heat followed by relaxation heat. This is the extra heat that is going to be produced in addition to the recovery heat where your muscle is restored to its previous length. Okay, so what is a fence effect? More work done causes more expenditure of energy and more heat produced. Okay, with this we are completing the thermal changes and all the properties. Then you need to understand what is summation of contraction. With this we can complete the properties. Summation means what? When the skeletal muscle is stimulated before its contraction, it gets over, it responds to another contraction. If it is repeatedly stimulated, the contraction summate without relaxation. Such a type of summated contraction is called as complete tetanus. Okay, complete tetanus. You call it as tetanus. Okay, during the complete tetanus, tetanus is not the disease. Here, complete tetanus means contraction. The tension developed is 4 times that developed in the individual twitch contraction. This is very important, guys. This carries one important MCQs in our test and discussion session. So what happens? Summation. So just consider this is going to be the force 1 that is going to be stimulated. That is going to cause contraction. There is going to be F2. There is going to be F3. Again and again, you're going to stimulate. Because of that, there is no relaxation at all. But when you give individually, so you're going to give muscle contraction, then you're going to relax, then you're going to give contraction, then you're going to relax. Then like this, when you're going to summate, that is at the end of the one contraction itself, you're going to give another stimulation, another contraction. Because of it, the contraction becomes 4 times. It twitches 4 times than that of the normal individual stimulus. Okay, the stimulation frequency at which summation of contraction occurs is determined by knowing the twitch duration of a muscle. What is tetanizing frequency? In the my frequency occur aguna, what is that? 1 divided by twitch duration. Okay, twitch duration. Evlo nero. At how many duration one muscle contracts, one muscle twitches, and the timing you can calculate the twitch frequency. For example, if a twitch duration is going to be 10 milliseconds, frequency is less than 1 by 10 milliseconds. Okay, 1 by 10 is nothing but 100 milliseconds. So when there is a frequency less than 100 milliseconds, this causes discrete response interruption, and frequencies greater than 100 milliseconds may cause tetanic contraction. Okay, in the 100 per second, okay, 100 per second, okay. So you have this 100, 1 by 10, nothing but 1 by 10 millisecond, that is going to be 100 per second. So 1 by 10 milliseconds kila, that what means that 100 per second, if you're going to give a okay twitches or stimulus, that can cause a greater tetanic contraction. This is what you call it a summation of contraction. This is one of the properties. Now, moving on towards the neuromuscular junction, the another important part, guys, at this point of time, I'd like to remind you that than MCQs, this neuromuscular junction is a very important exam question. I need everyone to concentrate at this point of time. Close all your textbooks, close everything, whatever you have in your hand, just close, just understand this concept, because neuromuscular junction, just this diagram itself is going to carry you importance. Separate short note question. This entire neuromuscular junction is carried as a essay question. Numerous MCQs are asked from this point of time. Therefore, I request you all to have utmost concentration. In just next half an hour, we will be completing the entire neuromuscular junction. Okay, now just consider this one particular diagram. Okay, this is asked as an exam question for you all. Just consider this diagram. You will be having a nerve ending. You will be having a muscle ending. This muscle ending has numerous channels. These nerve endings also have numerous channels. Okay, they are going to interact. They are going to give you a response, that is a muscle contraction. Okay, in order for a muscle to contract, you need to first activate your nerves, isn't it? First, your nerves must be activated. So, for a nerve to get activated, can you all remember what is the nerve threshold? At what point of time your nerve gets activated? Remember, guys, your nerves will be getting activated at plus 40 millivolt. Then only there will be action potential generated inside your nerve. Once there is a plus 40 millivolt, what happens is you are at plus 40 millivolt. At plus 40 millivolt, your terminal button of your nerve, axon terminal button of your nerve is going to have numerous vesicles filled with acetylcholine. There are numerous vesicles filled with acetylcholine. Just go along with me. Don't think anything. Now your nerve gets activated at plus 40 millivolt. Once your nerve gets activated, your nerve is going to stimulate. You're going to transfer that action potential to your terminal button through your myelins. So myelins are the one that is going to exponentiate your action potentials. It is going to take towards your terminal button. At this terminal button, you have a channel called as calcium channels. So voltage gated calcium channels. Apart from the voltage gated calcium channels, you have two other channels that is called as syntobrevin, syntaxin. You're going to have two important channels that is called as syntobrevin, syntobrevin, otherwise called as V-SNAR, otherwise called as V-SNAR. Then you're going to have syntaxin and SNAP25. This is going to be called as T-SNAR. This is going to be called as T-SNAR. You have a S-SNAR. Okay, you're going to have a V-SNAR. You're going to have a T-SNAR. Remember, apart from it, you're going to have a okay, so all these things where it will be, I'll tell you one by one. So you have a T, so you are going to have a V-SNAR, you're going to have a T-SNAR, you're going to have voltage gated calcium channel. Now, for now, you're going to remember these three things. So these three things, okay, are going to be present in the nerves. Now, we are not talking about muscle now. We are going to talk only about the nerve. I'll tell you what happens once, okay, once there is going to be a opening of the voltage gated calcium channel because of this plus 40 millivolt. Because of this plus 40 millivolt, what happens? There is going to be opening of the voltage gated calcium channels. The calcium which is present in the extracellular fluid are now entering into the nerve terminals through voltage gated calcium channels. So now your calcium is going to enter inside your, so it's going to enter inside your terminal button. Now, inside the terminal button, you have several vesicles of acetylcholine. You have vesicles of acetylcholine like this. Just remember. So, you're going to have vesicles of acetylcholine like this. Okay. You're going to have 3 vesicles. Now, remember. Okay. Or 2 vesicles. These vesicles, inside your vesicle, there is going to be acetylcholine. So, inside this, what you have? Acetylcholine. This vesicles will be having a receptor, or you can call it as the attaching receptor. That attaching receptor which is present in your acetylcholine, you call it as a V snare. You call it as V snare, or you can otherwise call it as synaptobrevin. Now clear what is synaptobrevin? Now what happens at the terminal button? At the terminal button, you also have certain receptors. Okay, you also have certain receptors. What is this receptors called as? T snare, or you otherwise called as syntaxin. T for terminal button, remember. So you have a V snare, you have a T snare. This V snare and T snare has to bind to each other. Remember, this V snare and T snare, yeah, it has to come like this. It has to come like this. And what happens is, okay, this has to come like this. Okay, this has to come like this and it has to get attached like this. In order to attach, you need to have the synaptobrevin and syntaxin combines together. Once this is going to combine together, what happens? Exocytosis occurs. So only when these two are going to be combined, there is going to be exocytosis. Once exocytosis occurs, what happens? This, okay, what happens? This molecule, okay, has to get out. So this molecule, what happens? They will be getting out to the outer aspect. This is going to be your, okay, here what happens? You will be having your muscles, and this place is your neuromuscular junction. And what is this? Motor end plate. There's going to be a motor end plate. Remember, you will be having, right? So this is going to be a T tubules. At the end of T tubule, you will be having the motor end plate, isn't it, guys? Which we saw earlier. So this place is called as your neuromuscular junction. Now acetylcholine has come out. Okay. Now what happens to the acetylcholine which has come out? Okay. In neuron, I'll once again repeat, calcium from ECF pushes the vesicle down to touch the axonal ending, except your renin and parathyroid hormone, even when the intracellular calcium is less. Okay, it is going to come inside the vesicle. Acetylcholine is there. You have an undersurface protein, synaptobrevin, that is going to be pushing towards your syntaxin, and synaptobrevin syntaxin fuses each other, finds a space for the release of neurotransmitter into the synaptic cleft. This is going to be the synaptic cleft. Now, since it has come to the synaptic cleft, what happens? At the same time, when the nerve is excited by plus 90, okay, when the nerve is excited to the plus 40 millivolt, at the same time, your muscles should also get excited. So that is when you will be having neuromuscular contraction. So your cells are going to be at rest. Okay, now what happens? Your muscle cells are going to be at rest minus 90 millivolt. Okay, now when there is going to be now what happens when the muscle at a relaxed state? What happens? There is going to be a acetylcholine that is present in your in entire area. Now you have more acetylcholine. Okay, now you're going to have more acetylcholine like this. This acetylcholine actually binds, this acetylcholine binds to the ligand gated sodium channel. So what happens? Here you're going to have ligand gated sodium channels which are closed like this. You have a ligand gated sodium channels. This acetylcholine, what it does is, first this acetylcholine comes and binds to the, okay, first your acetylcholine comes and binds towards your sodium channel. What is the sodium channel? Ligand gated sodium channel. Okay, now acetylcholine comes, binds to the receptor sites. So acetylcholine binds to the receptor sites at the motor end plate. This site is called as this is going to be called as ligand-gated sodium channel. Once it comes and activates your ligand-gated sodium channel, now what happens? There is going to be an influx of sodium. There is going to be an influx of sodium into your muscle fibers. So muscle fibers kulla sodium increase ஆகுது. அப்ப நார்மலா muscle வந்து 90 millivolt ல இருக்கு. When there is going to be influx of sodium, this is going to change to minus 55, minus 90 millivolts minus 55 millivolts. It gets converted. So at minus 55 millivolts, what happens is it is going to activate another sodium channel that is called as your voltage gated sodium channel. So you will be having another channel that is called as voltage gated sodium channel. Now what happens? Your voltage gated sodium channels are going to be activated. So sodium influx causes voltage gated calcium channels to open. Okay, sodium channels open ஆகுது. இந்த sodium channels open ஆகும்போது என்ன ஆகும்? இன்னும் further it reduces to minus 40 millivolt. Okay, son, plus 40 millivolt. From minus 55 millivolt, due to the influx of sodium, sodium potassium channel, due to influx of sodium, what happens? Now it is going to be increasing the positivity inside your muscle cell that reaches plus 40 millivolt. When there is a plus 40 millivolt, what happens? There is going to be the activation of your calcium channel. What calcium channel gets activated? What is the calcium channel? You call it as anyone? I told you already. Okay, what is the calcium channel called as? You're going to call it as dihydropyridine receptor, which is present inside your tubule. This dihydropyridine receptor are going to activate your ryanodine receptor. This is going to cause skeletal muscle contraction. Okay, first we discussed about the skeletal muscle contraction in the first part. Now, how the skeletal muscle contraction gets activated? Through your what? Through your neuromuscular junction. Now answer me. V-SNAR protein is your synaptobrevin. And what is your T-SNAR protein? Syntaxin and your SNAP25. Ligand-gated sodium channels are going to cause local potential. What is this local potential called as? Local potential called as motor end plate potential. Now sodium entry is going to be occurring through, this ligand-gated sodium channels are going to be acetylcholine receptors, which is present at the motor end plate. Through this motor end plate, their ligand-gated sodium channel, this is going to cause local potential. This local potential is otherwise called as motor end plate potential. This motor end plate potential causes opening of voltage gated sodium channel, which further depolarizes to the plus 40 millivolt and causes all the skeletal muscle contraction. This V-SNAR protein is the one that is blocked by your tetanus toxin. Your tetanus toxin blocks us which protein? V-SNAR protein. And T-SNAR protein is blocked by Clostridium botulinum, or otherwise called as botulinum toxin. So your V-SNAR is blocked by tetanus toxin. T-SNAR is blocked by botulinum toxin. Clear how this occurs? How this action of your Clostridium toxin occurs. Now you all know that this is going to be your spinal cord. You're going to have a gray matter and you're going to have a white matter. You're going to have a gray matter and going to have a white matter. Okay, so what happens is So you're going to have this is considered as humerus. Okay, then you're going to have another one that is going to be a radius and ulna. Remember like this. So you're going to have biceps brachii. You're going to have biceps brachii, which is going to be attached like this. Okay, now what happens? Whenever a nerve comes, acetylcholine comes and attaches, sits in the synaptic terminal. Okay, what happens is that is going to carry out this acetylcholine is going to carry out stimulus. It's going to carry out stimulus towards your muscle fiber. Who is going to carry the stimulus? Okay, that is going to be your muscle 1B. Okay, 2A. You're going to have 1A, 1B, 2A. All these muscle fibers are going to carry sensation. From there, what happens? There is going to be a muscle contract பண்ணணும்னு சொல்லணும்ல, அந்த contraction யார் கேரி பண்ணிட்டு வர போறாங்க? ஒரு nerve has to carry. அந்த nerve carry பண்ணிட்டு வந்தாதான் muscle contraction நடக்கும். So now, for example, I am going to give you a iron box. So that iron box is going to be, unfortunately, without knowing, it has touched to your skin. This skin, what it does, it is going to carry out sensation to your spinal cord. Reflex mechanism activates. Then the reflex mechanism activate பண்ணனும்னா, you are going to withdraw your hand. For withdrawing your hand, the muscle has to contract. Who is going to carry the nerve signals? Nerve signal is carried by A alpha. A alpha is going to carry. A alpha causes contraction. How it causes contraction? By releasing acetylcholine at the motor end plate. So in the acetylcholine motor end plate release ஆகணும்னா, who is going to be important part is going to be played by your V snare, T snare. When the V snare is blocked, what happens? There is going to be a tetanus toxin comes. This tetanus toxin, what it does is, tetanus toxin are going to be inhibiting your V snare. So V snare is inhibited. Because of this, what happens? There is going to be, இப்போ V snare inhibit பண்ணிட்டேன்னா என்ன ஆகும்? acetylcholine release ஆகாது. Okay, you're going to inhibit the interneurons. Okay, this is going to be causing இது. So இப்போ இந்த V snare வந்து உட்காரணும்னா, okay, so for this V snare to come and sit, understand very carefully, you're going to block your V snare. So initially இந்த V snare வந்து உட்காரணும்னா, இது inhibitory neurons தான் inhibit பண்ணுது. Okay, this is going to inhibit the inhibitory neurons. Okay, your tetanus toxin is going to block the V snare. But which V snare is blocked? Remember, it is going to block your V snare of your alpha, A alpha V snare inhibitory protein side is going to block. It is going to block your V snare is going to be your inhibitory interneurons. Your V snare is going to be inhibitory interneurons. Inhibitory interneurons. So when you are going to block this inhibitory interneurons by your tetanus toxin, it was going to cause a excessive contraction of muscle. When there is a excessive contraction of muscle, it is going to lead to spastic paralysis. Whereas your botulinum toxin is going to be blocking your T snare. It is going to block your T snare. This T snare in the A alpha neuron, there is no acetylcholine release at all. இங்க acetylcholine release ஆகும். எப்ப ஆகும்னா, ஏன்னா உங்களுக்கு V snare மட்டும்தான் ப்ராப்ளம் இருக்கு. V snare pushes it down. But the vesicles which are present nearby towards your T snare, that will be causing release of your acetylcholine, isn't it? And the release of acetylcholine can occur. Therefore, அதனாலதான் வந்து when there is a V snare block, even then small amount of acetylcholine which are present in the initial part will be released. That causes excessive muscle contraction, that is spastic paralysis. Whereas in botulinum toxin, what happens? And the release of acetylcholine will not be there. When there is no release of acetylcholine, what happens is there is going to be a flaccid paralysis. In botulinum toxin, it is going to cause flaccid paralysis. T snare causes exocytosis. Remember, this is what you need to understand. T snare causes exocytosis. V snare causes pushing of your acetylcholine vesicle towards your terminal button. Okay, therefore, even when there is a blockage of your V snare, your T snare will be functional. Therefore, the acetylcholine vesicles which are present by near to the motor, near to your synaptic terminal, it is going to be causing exocytosis. Therefore, there will be initial muscle contraction. That is why you call it as you're going to call it as a excessive contraction of muscle, that is spastic paralysis. Whereas in botulinum toxin, there won't be release of acetylcholine. Therefore, you call it as a flaccid paralysis. Hope you're clear about this concept. Once you're clear about this concept, so you know this entire thing I have just put down, whatever I thought, I have just put down as a words so that you can read later. Now you know what is Clostridium tetani. Clostridium tetani, always remember, it is going to cause spastic paralysis. It's going to cause spastic paralysis. And what is your botulinum toxin going to cause? Botulinum toxin is going to cause flaccid paralysis. Flaccid paralysis. Okay, with this, I wanted to talk about two important, very, very important, two important thing I want to talk about in this synaptic terminal, in this neuromuscular junction itself, that is skeletal muscle relaxants. So whatever you read, try to make it as a clinical based, because that is what you are expected by the NMC curriculum, NMC pattern. Okay, so all these order of arrangements, you know that first this step has to occur, then calcium channel, then voltage gated calcium channel, then synaptobrevin, synaptoxin, then it has to, vesicle is going to be exocytose, then there is going to be a ligand gated sodium channel, then voltage gated sodium channel, then the calcium channel. The regulation yaar pandrangana, one small molecule that is called as neurexin. Okay, the small molecule called as neurexin is the one that is going to do the orderly arrangement. Okay, this is going to do orderly arrangement. This is what I need to remember, you guys. So now, what are the skeletal muscle relaxants? Neuromuscular transmission blockers are frequently used as a muscle relaxants during surgical anesthesia. Two groups of skeletal muscle relaxants are available. One group blocks the binding of acetylcholine to its receptor, thereby preventing depolarization. They are called as competitive muscle relaxants. So in the area and the ligand gated sodium channels, it binds. So what are the drugs that binds with the ligand gated sodium channels? So ligand gated sodium channels la poi bind panni block pandra drugs. Okay, blockers வந்து யாரான்னு பார்த்தோம்னா all curarine drugs, all curium drugs. Okay, tubacurarine, pancuronium, then you're going to have your cistrocurium, then you have this vicuronium, gentacurium, curonium, curium, curine. In the எல்லா drugsமே they are non-depolarizing muscle relaxants. There are other group of drugs that acts as an agonist to your acetylcholine receptor. So that is going to act as an acetylcholine receptor agonist பண்ணும் and causes sustained depolarization. When you're going to have a sustained depolarization, that will be causing prolonged flaccid paralysis. This is the non-competitive depolarizing muscle relaxants. Example is going to be your succinylcholine. Agonist act பண்ணி block பண்றவங்க யாரு? Succinylcholine. Okay, I want the first year itself to remember these questions, because whatever you're reading it now, along with physiology, will be retaining in your mind. Even though you will be reading this in the pharmacology, you will read about the drug name, adverse effect, uses, everything. Now you're going to simply remember which binds with the sodium blockers and blocks it, all the curonium drugs, all curium drugs, all curarine drugs, and which is going to agonist act பண்ண போது succinylcholine. Clear? Okay, so acetylcholine esterase inhibitor. Now with this, I wanted to add one more important point. Okay, there will be excessive acetylcholine here. These excessive acetylcholine will be destroyed. Why? Because you don't need a prolonged action. Who is going to destroy it? By the acetylcholine esterase. They are going to destroy it. So this drug, who is going to be these drugs? So normally acetylcholine esterase will be there to destroy. There are some drugs by which you can prevent the neuromuscular junction, thereby blocking the muscle contraction. Okay, first thing, you can either go for like sodium channel binding blockers, or you can go for sodium channel agonist, or you can also go for acetylcholine esterase inhibitor. What are the drugs? Okay, so you have this edrophonium, physostigmine, neostigmine, ecothiophyte, all these things. I think I already, if you want to learn more, guys, if you want to learn about this neuromuscular blocking more, can go and read now itself. In our channel, we have the drugs called as anticholinergic drugs and cholinergic drugs. இந்த ரெண்டு சாப்டர் இருக்கு, cholinergic, anticholinergic. இதுல cholinergic drugsல you can see this acetylcholine esterase. And acetylcholine esterase எப்படி inhibit பண்ண போறீங்க? By acetylcholine esterase inhibitor. Where you have this application, applied physiology. This is what you're going to apply in a poison in case of a farmer who was going to take fertilizers, pesticides, and they are going to be taking that, okay, they're consuming that, okay, and ingesting that, they will be coming to you. Okay, that is called as your, okay, pesticide, insecticide. So they will be using this edrophonium. Okay, they will be using this acetylcholine esterase. Okay, that point of time, what you will be doing is you will be giving acetylcholine esterase inhibitors. So that is your edrophonium. So all these things are drugs there. So that in that case, you will be giving acetylcholine esterase inhibitors. Okay, so that is what you need to understand here. That is also used as a neuromuscular blocker. Now various other numerous blockers. Okay, so if you see in a mamba snake, okay, sometime what happens, it gives you dendrotoxin. This dendrotoxin, what it does is it will be inhibiting the voltage gated channels. Voltage gated channels inhibit பண்ணுச்சுனா அப்ப என்ன ஆகும்? That is again going to cause a problem. Similarly, you have this calcium channel, isn't it? Voltage gated calcium channels. At the starting I told you, right? This voltage gated calcium channels are inhibited by the toxin which is present in the snail. That is conotoxin. That conotoxin is going to block this particular area. Conotoxin blocks this particular area. That is very important. Okay, conotoxin blocks this particular area, that is voltage gated calcium channels. So when there is going to be blockage of voltage gated sodium channels, that is going to be your tetrodotoxin. So this is present in the puffer fish. All these things are extra points, guys. You can remember or you don't want to remember, that's up to you. Okay, now moving on towards the next important thing, that is going to be two important diseases you are going to see. One is your myasthenia gravis and second is a Lambton. Okay, we are going to see the second disease, that is going to be your Lambert-Eaton myasthenia syndrome. Okay, what is myasthenia gravis? Fatal disease in which skeletal muscles are going to be weak and they are going to tire immediately. Why this occurs? This occurs due to the fatal disease. It is an autoimmune disease which is mediated by a specific antibody against this acetylcholine receptors. Okay, so what happens is it is going to be binding to the nicotinic acetylcholine receptors. Whenever they are going to bind with the receptors, acetylcholine receptors, which are present in the postsynaptic area, that is going to cause destruction. So their muscles won't be able to contract. That time what happens, there is going to be weakness of muscle. So because of this weakness of muscle, so இது வந்து இந்த weakness of muscle இருந்தாலும் இது என்ன டைப்பா இருக்கும்னு பாத்தீங்கன்னா flaccid type of paralysis. Why I call it as a plastic type of paralysis? So flaccid இருக்கும், plus என்னன்னா இங்க weakness இருக்கு. So அப்ப எங்கெங்கெல்லாம் இருக்கும், major symptoms எங்கெங்கன்னா ptosis will be there. Second thing, dysarthria, dysphagia, upper limb weakness, respiratory muscles will be affected rarely. So what happens is with repeated activities, now what happens when the patient will be normal muscle power will be there after sleep and rest. Okay, when you're going to give a repeated activity, what happens? Already there is going to be a blockage of your acetylcholine receptors. Already there is a blockage of your acetylcholine receptors. Remember carefully. Okay, now you're going to increase the muscle contraction, muscle firing, you're going to increase. But there is not going to be weakness is there already. Already there is going to be less acetylcholine that is entering because of the blockage of your acetylcholine receptors. Now again, what you're going to do, you're going to increase the activity that will be worsening your symptoms. So it's autoantibody, thyrotoxicosin, thioma, okay, all these things can be differential diagnosis. Now what you will be doing is here, you will be giving neostigmine. Okay, you will be giving neostigmine challenging test. Neostigmine is going to be acetylcholine esterase inhibitor. When you're going to give acetylcholine esterase inhibitor, this is going to increase the concentration of acetylcholine. When more acetylcholine is available by some point of way, so by some receptors, there is a chance of, okay, contraction. So when they are going to give more amount of acetylcholine, contraction can occur. So that is some chances are there. So that is called as neostigmine challenge test. But even though you give more neostigmine, that is not going to be useful. Why? Because receptors are going to be remaining the same number. Receptors are the one that are going to be affected. Even if you give more amount of acetylcholine, okay, even when there is more amount of concentration of acetylcholine also, it cannot bind. Okay, that is called as all or none phenomena. Either the receptors can activate or receptors cannot be activated. Okay, this is all about your myasthenia gravis. Okay, what is going to be a treatment? You can give physostigmine, corticosteroids to suppress the antibodies, you can have immunosuppressive drugs. So all these things are aimed to inhibit the antibodies that are formed against your receptors. Okay, then you can give intravenous immunoglobulin, or you can go thymectomy, the thyma, which is going to be thymoma, that is going to cause autoantibodies. Okay, now coming to the Lambert-Eaton syndrome. Lambert-Eaton syndrome, your autoantibodies are produced against your calcium channels in the neuronal ending. Okay, neuronal ending calcium channels. Example, can you tell something? So the same way the calcium channels are blocked, okay, by some drug. You have read about some drug that blocks the calcium channels. What is that drug? Okay, it's a toxin that is going to be present in the snail, that is canotoxin. Similar to canotoxin is your antibody that are going to be acting against your calcium channels in the presynaptic membrane. Therefore, calcium influx is reduced, acetylcholine release is reduced, and there is going to be muscular weakness. In this type, if you're going to give repeated activity, more amount of calcium action potential is generated. Because of more amount of action potential, new receptors can be made. Your body senses and creates new calcium channels, and because of that, they can be for a short period of time, there can be improvement of movement. And example is going to be your small cell lung cancer, that is going to be also having a similar antibodies produced, and it is going to be Lambert-Eaton syndrome. This is called as paraneoplastic syndromes, right? So paraneoplastic syndrome in your small cell lung cancer. Now along with me, write this tabular column. So pathophysiology, what happens here? Antibody mediated post-synaptic acetylcholine receptor blockage. Okay, what happens in your Lambert-Eaton syndrome? So there is going to be antibody paraneoplastic that is going to be blocking your or inhibiting your calcium receptors that are present in the presynaptic terminal. Okay, because of this, what happens? Because of the myasthenia gravis, there is going to be weakening of muscle, causes ptosis, dysarthria, then dysphagia, then there can be upper limb weakness. Okay, immediate presenting feature. Whereas in a Lambert-Eaton syndrome, what happens? There is going to be weakness of muscle, but there is some exception. So there are some muscles that are spared. Example, intraocular muscles are spared in this, bulbar muscles are spared in this, limb muscles are spared in this. Limb muscles are going to be spared in this. With repeated activity, there is going to be worsening of your symptom. Whereas in Lambert-Eaton syndrome, there is a improvement. There can be improvement of the symptom. Okay, improvement of the symptom most commonly associated with thioma, This is most commonly associated with small cell lung cancer, small cell lung cancer. Okay, then what is the treatment here? Treatment here is going to be anticholinesterase drug along with the glucocorticoids, cyclosporin, azothioprine, rituximab. Whereas Lambert and Eaton, you can give pyridostigmine and diaminopyridine. This diaminopyridine acts by blocking the potassium channel, which results in prolonged depolarization of motor nerve terminals, and this enhances the acetylcholine release. Short term or treatment மாதிரி நம்ம இதை பண்ணலாம். Okay, so this is importance of the entire thing. Again, along with this, I also wanted to emphasize what rigor mortis. So இப்போ எல்லாமே integrate பண்றாங்க. So rigor mortis is a forensic medicine question. But again, you can integrate that inside your this physiology itself. When ATP is not available in a dead body, ATP is not available, your myosin head is going to continue to bind with the active side of your actin via adenosine diphosphate, and your muscle is unable to relax. Such sustained contraction is what you're going to see after the death. This is called as postmortem stiffening or cadaveric rigidity. Rigor mortis mostly helps you to know the same time since death, because in general, apparently 1 to 2 hours after death, there gets a well-established rigor mortis in about 9 to 12 hours. It is maintained for 12 hours, then gradually pass in the 12 hours. This 12 hours rule, first 12 hours, contraction, maintenance for next 12 hours, then disappears in the next 12 hours. According to the Nisten's rule, cadaveric rigidity affects successively the masticatory muscle, that is your jaw, and those of the face, neck, then trunk, arm, then finally those of the lower limb. And when there is a disappearance of rigor, it follows the same pattern. It follows the same pattern. Now you know what are the things. So now I want to talk about the drugs that are going to be causing neuromuscular transmission. I think we have already discussed this depolarizing drug and your in the drug. Both I think I have discussed. So okay, that's all. I don't want to go more further on this point. So depolarizing drug, non-depolarizing drug, you need to understand both the thing. Depolarizing drug or adverse effect, succinylcholine or adverse effect என்னன்னா, it is going to be cause respiratory depression and malignant hyperthermia. But remember, depolarizing drugs are shorter duration of action, only 3 to 5 minutes. Whereas non-depolarizing like atracurium, vicurium, all these things are going to have a longer duration of action. Bronchospasm and hypotension are the common side effects. Now moving on towards the very important thing, that are the types of your muscle fibers. Remember, you have two important types, type 1, type 2. Okay, type 1 are those, okay, very small sized fibers. They are very less in number in your myofibril. They are going to be small sized. They are very less in number. Okay, wow. So this causes less contraction. Force of contraction, power is going to be very low. It takes more time or slow time to begin contraction. So time it is going to be very slow. Hence you call it as slow twitch fibers or slow acting fibers. But remember, even though slow timing, so time slow andalo, the time when acting duration, when you're going to consider duration of action, it is going to be very long. Duration of action is going to be very long. Clear? So duration of action is going to be very long. They are very resistant to fatigue. They are very resistant to fatigue. ATP is produced by aerobic metabolism. So aerobic glycolysis, oxidation of fatty acid. So it has a very high oxidative capacity. Okay, so enormously increased, you're going to have a mitochondria to synthesis, a high density of mitochondria is there to synthesis the capillaries to supply oxygen. There will be huge density of capillary to supply oxygen. Therefore, these fibers are called as, otherwise called as red fibers. So, otherwise called as red fibers. So, what is the other name of this? So, this is otherwise called as red fibers. Okay? Red fibers. Otherwise, okay, also called as, okay, it's going to be also called as red fibers. Clear? Now coming towards your type 2. Type 2 is going to be large cells or sized cells, very large, and they're going to have increased myofibril. Force of contractions will be increased. So fast twitching fibers, you call it as a fast twitching fibers, short duration of action, but duration of action will be short. Okay, they are very prone to fatigueness. So fatigueness is very prone. Okay, only glyconeogenesis by ATP. So therefore there is an anaerobic glycolysis. glycolysis. ரெண்டுமே anaerobic glycolysis. Very less number of mitochondria is there, very less capillary density will be there, very less myoglobins are produced, and then increase space for storage of glycogen. Storage fuel வந்து glycogen store பண்ண முடியும். ஓகேவா? So this is otherwise called as white fibers. This is otherwise called as white fibers. This is what you need to understand. Okay, again type 2 fibers are two type 2A, 2B. So that is what I have mentioned here. You can read if you want, otherwise there is not needed. So now coming to the difference between skeletal muscles and smooth muscles. Now understand that you have a skeletal muscle, you have a smooth muscle. So you have a smooth muscle estimated. Now the smooth muscle வந்து பாத்தீங்கன்னா, what is the difference? What is the difference for your smooth muscle? So I'll just draw a tableau column. We will discuss it. So this is going to be a skeletal muscle, this is going to be a smooth muscle. Remember, in smooth muscle there is no T tubules that is present. So absence of your T tubule, absence of T tubules. Okay, whereas அங்க நம்ம T tubules இருக்குன்னு பார்த்தோமா, இங்க T tubules இருக்காது. Number 1. Okay, number 2 is you are going to have, what is that? So there is going to be a motor end plate, and the motor end plate will not be there. So in smooth muscles there won't be motor end plate. Number 2. Okay, now you don't have a, if you don't have a motor end plate, there will be no neuromuscular junction. Instead of neuromuscular junction, it is going to be replaced by, it's going to be replaced by neuron varicosity. Varicosity. This is going to be your third point. So first one, there is a presence of T tubules. There is a presence of T tubules. Number 2, there is a presence of motor end plate. Number 3, there is a presence of neuromuscular junction. There is a presence of neuromuscular junction. Number 4, okay, this doesn't have myofibrils. So myofibrils la da sarcomere இருக்கும். அப்ப myofibrils இல்ல. அப்ப myofibril la da Z line present ஆயிருக்கும். அப்ப there is no Z line. There is no Z line. So that is the next thing you are going to remember. There is no Z line. That is the next thing you are going to remember. So I am going to talk about smooth muscle here. Okay, I'm going to talk about skeletal muscle here. Okay, there is no Z line. So if there is no Z line, where is the thick filament, thin filament? Remember, your thin filament is going to be hooking to the dense body, hooked to the dense bodies. And where is your thick filament? So thick filament is going to be present in the cytoplasm. Your thick filament is present in the cytoplasm. So because of this, there is no M line, there is no Z line, there is no M line. Okay, then your calcium, you will be having a calcium, right? This calcium does not combine with tropsy. So the calcium combining with tropsy in the concept line illa. So there is no combination of calcium and tropsy. In fact, solapona, there is absence of tropsy. There is no tropsy present in your smooth muscles. Okay, this tropsy is replaced by, a structure called as calmadulin. Okay, this is another question which I kept in the test and discussion. So absence of tropsy, this is replaced by calmadulin. Okay, and then now coming back to the resting membrane potential. Okay, resting membrane potential. Okay, remember, resting membrane potential, this is going to be a smooth muscle, this is going to be a skeletal muscle. The resting membrane potential of your smooth muscle is going to be minus 60 millivolt to minus 50 millivolt. Okay, and then it is also requires less ATP. It requires less ATP. Okay, that is why this is called as, this is done by latch bridge mechanism. Latch bridge mechanism. I'll discuss about this latch bridge mechanism now. Okay, this is called as latch bridge mechanism. Clear? So ATP less ada required iruko. Okay, then how does the depolarization occurs? There is no sodium influx at all. Only calcium influx is present. Only calcium influx is present. Okay, I have discussed 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 points. Or essay keta kuda you will be able to write now. Okay, so இதுக்கு எல்லாமே exactly opposite pa. So no Z line present, myofibrils present, your thin filament is attached to the Z line, your thick filament is attached to the M line, then you have the trop C is going to be attached to the calcium, and you have resting membrane potential minus 90 millivolt, minus 90 millivolt. So in your trop C is going to be present, your thick filament is going to be attached with your M line, and you have thin filament that is going to be attached with your Z line. So there is Z line is going to be present. You're going to have myofibrils. Okay, myofibrils are present. So all this you're going to put down. Okay, then ATP is requires ATP for contraction. Even for relaxation, ATP is required. So relaxation kuda you need ATP. ATP is needed. Okay, that's all. This is the difference between the smooth muscle cells and skeletal muscle cells. Now coming back, the smooth muscle cells does not have a tubules, no motor end plate, very little sarcoplasmic reticulum, no myofibril. Because of all these things, you don't have a motor junction also. Then what happens is there is going to be a dense bodies. Can you see here? In the dense bodies la da ungaloda in the line your thin filaments will be attached. So this is your thin filament, this is your thick filament. Can you see here? Your thin filament is attached by dense bodies. Your myosin is going to slide along. So the actin myosin are loosely arranged. Okay, myosin act along the long distance. Smooth muscle myosin is inched along its head. So in the calcium அப்படிங்கிறது poorly developed sarcoplasmic reticulum. அப்ப இந்த calcium poorly developed இருக்குன்னா smooth muscle contraction is highly dependent on the extracellular calcium concentration. அப்ப extracellular calcium வெளில வரணும்னா அதுக்கு you need a influx of calcium through a voltage gated calcium channels. Once there is a voltage gated calcium channels, your calcium comes inside and calcium is going to bind to the calcium calmodulin. Okay, you call it as calmodulin, and the calmodulin la bind achna, it activates your myosin light chain kinase, MLCK. Myosin light chain kinase enna aguna, there is a phosphorylation of myosin using energy and phosphate from ATP. This binding of myosin to actin and sliding of actin over myosin causes contraction of your smooth muscle. I think you can take the screenshot of this particular slide which acts everything. So then how it relaxes? இதுக்கு ATP எல்லாம் தேவையில்லை. Dephosphorylation of myosin by myosin light chain phosphate. Calcium is removed from the cytoplasm via calcium sodium antipore. So calmodulin releases calcium and myosin light chain kinase is phosphorylated and there is going to be relaxation. This is going to cause relaxation. Is it clear? So now you understood how there is going to be relaxation also. Now you understood how there is going to be relaxation also. I think everything is clear up to this. Okay, I think everything is going to be clear up to this. Okay, next what happens is, okay, so and I told you a latching system, isn't it? So prolonged tonic contraction due to the myosin cross bridges attaching with lactin is called as latch system. So this does not spend energy. There is a continued excitatory signal. So calcium calmodulin dissociates only when relaxation occurs. Calcium calmodulin is going to be dissociating. Okay, so this is going to be done. Okay, is it clear? So urinary bladder full of urine, that time what happens? There is going to be a contraction. So that is what exactly occurs. So now you know the difference between the smooth muscle. I think you can also take a screenshot of this. Okay, so now what is a cardiac muscle? Now last and finally, you have one important thing, that is cardiac muscle. Okay, remember your cardiac muscles are going to be striated like your skeletal muscle, but the difference is in the contraction is going to be involuntary in nature. So the defining feature of cardiac muscle is going to have an intercalated disc and they have a gap junction that allows rapid diffusion of ion. Because of this gap junction, what happens? Your muscles is it's going to be syncing. Okay, there's going to be syncing with each other. மீதி இருக்கிற cardiac muscle ஓட எல்லாமே we will be discussing in the cardiac muscle part. Okay, so with this actually we are completing the entire muscle physiology. In the next part we will discuss about the nerve physiology. Once your muscle physiology, nerve physiology gets over, next we will be going towards the neurophysiology. That is going to be a CNS system which we will be discussing later. So thank you so much. Have a good day.